Cellular components of the cutaneous immune system. Introduction. The skin is the body's largest organ and a crucial protective barrier. It hosts a complex cellular immune network that defends against microbes and other stressors. This network includes skin-resident immune cells and circulating lymphocytes. Parenchymal cells like keratinocytes, endothelial cells, and fibroblasts also participate. The system is a complex web where cell types have both specific and overlapping roles. Innate versus adaptive immunity. Innate immune system is the rapid response, the first line of defense. Recognition relies on germline-encoded pattern recognition receptors, PRRs, to recognize broad classes of pathogens. No memory in innate immunity. Primary cells include dendritic cells and macrophages, mast cells, neutrophils, and resident tissue cells. Adaptive immune system. Slow response, takes time to develop. Specificity, uses somatic recombination to generate a vast diversity of antigen receptors. Memory, creates long-lived, antigen-specific lymphocytes for a more rapid and robust response upon re-exposure. Primary cells, T cells and B cells. Overlap, some cells like innate lymphoid cells, ILCs, and innate-like T cells, have characteristics of both systems. Components of the cellular immune system in healthy skin. Multiple different immune cell populations are present in healthy skin. Langerhans cells and resident memory T cells, primarily CD8+, reside in the epidermal layer, whereas CD4+ and T cells are found in the upper dermis. Innate lymphoid cells, ILCs, are found in proximity to the dermal-epidermal junction, whereas mast cells are often in proximity to dermal blood vessels. Dermal dendritic cells and macrophages are found in the dermis. Other cells may contribute to immune responses in skin such as fibroblasts, lymphatic blood vessels, nerves, and melanocytes. The immune response. The innate and adaptive systems work together. The innate response provides immediate defense and instructs the adaptive response on what type of reaction to mount. Steps of a response. A foreign pathogen enters. Innate response is activated, rapid phagocytosis and antimicrobial activity. Innate cells produce cytokines and costimulatory molecules that activate the adaptive system. Adaptive response is activated. T and B cells undergo gene rearrangement and clonal expansion. The immune system generates memory cells for future defense. Innate and adaptive cells. Interstitial lymphoid cells and innate-like T cells have both innate and adaptive immune functions. The immune system of higher vertebrates uses both innate and adaptive immune responses. These immune responses differ in the way they recognize foreign antigens and the speed with which they respond, yet they complement each other in eradicating foreign pathogens. T cells. Origin, derived from hematopoietic stem cells in the bone marrow and mature in the thymus. Major lineages, alpha-beta T cells, the majority of T cells. Gamma-delta T cells, a smaller subset with limited diversity considered part of the innate immune system. Naive T cells, circulate in the bloodstream until they encounter a specific antigen. Differentiation, upon activation. They differentiate into effector T cells, mount an immediate response. Memory T cells provide long-term immunity. T cell subsets. Based on coreceptor expression, CD8+ T cells recognize antigens on MHC class I molecules. They differentiate into cytotoxic T cells that kill infected cells. CD4+ T cells recognize antigens on MHC class II molecules. They differentiate into various helper subsets. Key helper subsets. T helper 1, Th1 produce IFN gamma and activate macrophages to fight intracellular pathogens. T helper 2, Th2 produce IL4, IL5, and IL13 and are involved in allergic reactions and defense against parasites. T helper 17, Th17 produce IL-17 and are crucial for fighting extracellular bacteria and fungi. T regulatory cells, Tregs suppress the immune response to maintain tolerance and prevent autoimmunity. Schematic view of events governing and occurring in T-cell differentiation. Depending on the type and activation status of the antigen-presenting dendritic cells, DCs, and on the type and amounts of cytokines secreted by these and/or other cells, naive T cells will expand and differentiate into various subsets, that is, Th1, Th2, Th9, Th17, Th22, Treg, and TFH. They exhibit different types of transcription factors and secrete different types of cytokines. Contributions of T effector memory, central memory, and resident memory cells to skin immunity. After exposure to bacterial pathogens, resident dendritic cells in the skin become activated, i.e., through activation of toll-like receptors, TLR, and migrate to skin-draining lymph nodes. Naive T cells recognizing bacterial peptide antigens on the surface of dendritic cells differentiate and polarize into skin-homing effector memory T cells, TEM, and central memory T cells, TCM. Skin-homing TEM enter all areas of the skin, but in highest number at the site of pathogen exposure. These cells will contribute to pathogen clearance, and some will become long-lived tissue-resident memory T cells, TRM. T cells are also released from the skin-draining lymph nodes and migrate to secondary lymphoid tissue, including non-cutaneous lymph nodes, giving rise to new populations of TEM that home to other non-skin peripheral tissues such as the gut and lungs. B cells. Primary function. B cells are lymphocytes responsible for humoral immunity by producing antibodies. Antibodies protect against extracellular pathogens through neutralization, opsonization, and complement activation. Development and distribution. B cells develop and mature in the bone marrow. After maturation, they migrate to secondary lymphoid organs, spleen, lymph nodes, and gut-associated lymphoid tissue, GALT. Activation. Most B cell responses require help from T follicular helper cells. Following activation, B cells differentiate into plasma cells and memory B cells. Cytokines produced by T cells influence which antibody class A B cell produces. IL-4 promotes switching to IgG1, IgG4, and IgE. IFN gamma promotes switching to IgG3. TGF-beta promotes switching to IgA, the major antibody involved in mucosal immunity. Additional functions. Besides antibody production, B cells also present antigens to T cells via MHC class II molecules, helping activate adaptive immune responses. Secrete cytokines. B cells in the skin. Under normal homeostatic conditions, B cells are rare in healthy skin, and it remains uncertain whether they are true tissue-resident cells or transient circulating cells. B cells become more abundant in chronic inflammatory skin diseases including lupus, leishmaniasis, and other chronic inflammatory dermatoses. A specialized subset called regulatory B cells, BREGs, produces IL-10, an anti-inflammatory cytokine. IL-10 BREGs help suppress excessive immune responses and maintain immune balance. Their presence and immunoregulatory role in human skin have only been described relatively recently and remain an active area of research. Unconventional and innate like T cells and innate lymphoid cells ILCs. In recent years, a number of other non-recirculating lymphocyte populations have been characterized. These lymphocyte populations are residents of non-lymphoid tissue and organs and include invariant natural killer T (iNKT) cells, mucosal-associated invariant T (MAIT) cells, T cells, and intestinal intraepithelial lymphocytes (IELs), as well as the emerging family of ILCs. These tissue-resident lymphocytes span the continuum between the innate and adaptive immune system and share a number of features pertaining to their tissue-resident functions. It is thought that these cell populations act as sensors of perturbed tissue integrity such as from infection or injury and help recruit additional immune cells from blood, as well as amplifying local inflammatory responses through their effect on parenchymal cells. Generation and effector cytokines of innate lymphoid cells. 3 populations of ILCs are found in human skin, ILC1 to 3. ILC1 express the surface molecule CD161 and are thought to be activated by IL-12 and IL-15. The primary cytokines produced by ILC1s are TNF and IFN. ILC1 are implicated in the pathogenesis of allergic contact dermatitis. ILC2 are activated by the Th2 cytokines IL-25 and IL-33, TSLP, and prostaglandin PGD2. ILC2 express the receptor KLRG1 and CRTH2, CCR4, and receptors for IL-25, IL-33, and TSLP. The main effector cytokines from ILC2 include LI5 and IL-13, and these cells have been implicated in the pathogenesis of atopic dermatitis. ILC3 are activated by IL-1 and IL-23, express the receptor NKP44, and produce IL-17 and IL-22. They are implicated in the pathogenesis of psoriasis. Each ILC subset is characterized by expression of transcription factors in similar manner as Th subsets. Thus, similar to Th1 cells, ILC1 express Tbet, ILC2 similar to Th2 cells express GATA3 and ILC3 express RORT similar to Th17 cells. Unconventional T-cell populations. Unconventional T cells respond more quickly compared with their conventional counterparts, CD8 and CD4 T cells. These cells are characterized by their limited TCR rearrangement. However, they expand the repertoire of antigens recognized by T cells. Thus, invariant natural killer INK T cells are able to respond to lipid antigens presented by CD1D. Other T-cell subsets including and T cells are also able to recognize lipid antigens bound by members of the CD1 family of proteins. T cells are able to recognize a diverse array of structures including phosphoantigens and sphingolipids apparently independent of MHC antigen presentation. Mucosal-associated invariant T MAIT cells recognize antigens presented in the context of an MHC-like molecule, MR1, on the surface of antigen-presenting cells or epithelia. MAIT cells respond to antigens derived from the structures of vitamins B2 and B9 containing a ribitol carbohydrate group. Natural killer NK cells. Natural killer cells appear as large granular lymphocytes. These cells are characterized by being CD16 plus CD56 plus CD94 plus CD161 plus, but negative for the T-cell markers CD3. NK cells have a role in surveying the body, looking for altered cells, either transformed or infected with viruses or parasites. The cells harboring these pathogens are killed directly via perforin, granzyme, granulysin, or Fos ligand-dependent mechanisms, or indirectly via the secretion of cytokines.